Welcome to Repro Decoded. In vitro fertilization, or IVF, is one of the most advanced interventions in reproductive medicine. However, despite precise clinical protocols, many cycles do not result in a successful pregnancy. For medical students and clinicians, understanding IVF failure requires looking past the clinical steps and diving into the exact cellular, genetic, and biochemical barriers to development. In this lecture, we decode the primary academic reasons why IVF cycles fail: oocyte quality and aneuploidy. The most common cause of IVF failure, particularly in advanced maternal age, is embryonic aneuploidy, or an abnormal number of chromosomes. This issue typically traces back to defects in the maternal oocyte. As an oocyte matures and resumes meiosis, it relies on a delicate cellular structure known as the meiotic spindle apparatus to pull chromosomes apart equally. With age or due to environmental stress, this spindle apparatus can degrade or become unstable. This leads to non-disjunction, where chromosomes fail to separate correctly. When fertilized, these oocytes produce embryos with structural genetic abnormalities. While these embryos may look completely normal under a lab microscope during a standard morphology grading, the underlying genetic defects almost always lead to developmental arrest before or shortly after transfer. Fertilization and egg activation. Failure. Even when high-quality oocytes and sperm are retrieved, fertilization is never guaranteed. In standard IVF, sperm may fail to penetrate the cumulus oophorus or bind to the ZP3 receptors of the Zona pellucida. To overcome this, clinicians often utilize intracytoplasmic sperm injection, or ICSI, where a single spermatozoon is injected directly into the oocyte cytoplasm. Yet, even with ICSI, fertilization can still fail due to a lack of oocyte activation. Normally, sperm entry must trigger a series of intracellular calcium oscillations within the egg. If the sperm lacks the specific enzyme, phospholipase C zeta, or if the oocyte's internal endoplasmic reticulum stores cannot release enough calcium, the oocyte remains unactivated. Without this calcium wave, the egg cannot complete meiosis II, the pronuclei never fuse, and the cycle stalls at day 1, embryonic genome activation and arrest. If fertilization is successful, the zygote begins early cleavage divisions. However, a significant percentage of embryos undergo developmental arrest between day 3 and day 5, failing to reach the crucial blastocyst stage. This milestone represents a major biological hurdle known as embryonic genome activation. For the first 3 days of division, the embryo is driven entirely by maternal RNA and proteins stored inside the original egg. By the 4 to 8 cell stage, the embryo must successfully switch on its own newly combined paternal and maternal genome. If there are underlying transcription defects, severe metabolic flaws, or mitochondrial insufficiency within the blastomers, the embryo cannot activate its own DNA. It runs out of maternal cellular energy and permanently arrests in the dish. Altered endometrial receptivity. An IVF cycle can also fail even if a genetically perfect euploid blastocyst is transferred. This shifts the focus from the embryo to the maternal uterine environment, specifically the endometrium. For successful implantation, the blastocyst must land within a highly restrictive time frame known as the window of implantation, which typically occurs during the mid-secretory phase of the menstrual cycle. This window is strictly regulated by progesterone. In fresh IVF cycles, high-dose ovarian stimulation medications can cause estrogen levels to skyrocket abnormally. This hormonal spike can prematurely advance the endometrial maturation, closing the window before the embryo is even transferred. Without perfect synchronization, the endometrium will fail to express essential adhesion molecules, such as integrins and pinopodies, causing the blastocyst to slide off the uterine wall without implanting. Hatching and microenvironmental factors. The final barrier to a successful IVF cycle occurs at the physical point of contact during implantation. Before a blastocyst can adhere to the uterine wall, it must successfully hatch by breaching and shedding its outer glycoprotein shell, the Zona pellucida. In an IVF lab, the artificial culture media or the freezing and thawing processes used during a frozen embryo transfer FET, can sometimes cause the zona pellucida to prematurely harden. If the blastocyst cannot cellularly digest its way out of this hardened shell, it remains trapped and cannot make direct contact with the maternal endometrium. Furthermore, localized microenvironmental uterine factors, such as chronic endometritis, silent endometriosis, or anatomical uterine septums, can create a highly inflammatory, localized immune response. This hostile environment causes maternal natural killer cells and macrophages to reject the blastocyst at the landing site. Outro. In conclusion, IVF failure is rarely caused by a single clinical mistake. Instead, it is a complex intersection of meiotic errors, egg activation failure, embryonic genome arrest, and anatomical desynchronization. Decoding these specific cellular milestones allows reproductive endocrinologists to adjust protocols, such as implementing pre-implantation genetic testing, PGTA, or utilizing artificial oocyte activation to improve outcomes in future cycles. Thank you for joining us on this clinical review. If this detailed academic breakdown helps your medical studies or clinical understanding, please hit the like button. Subscribe to